Hey guys, hope you're all doing well. Welcome to the fifth week of the semester. This week we will be covering Chapter 8, which covers cellular division, which is a form of asexual reproduction. Basically, all of the cells that are produced from the parent cell are going to be identical to the parent cell. So skin cells are going to produce skin cells. Particular tissues will produce tissue cells, bone cells will produce bone cells, so on and so forth. Now, the main point of cellular reproduction is going to be replacing any worn out or dead cells that we may have. Cellular reproduction is very important for any multicellular organism, and humans have trillions of cells because of cellular reproduction. Cellular reproduction is the main reason we are alive today. Through cellular reproduction, we developed into adults and are able to sustain healthy organs and tissues. All cells start off as stem cells, but as soon as they differentiate into specialized cells being like white blood cells, red blood cells you would find in the immune system, the various types of muscle cells that we have, or skin cells, that is what I mean by specialization. Like I mentioned earlier, they use asexual reproduction to continue creating these new cells. Cell reproduction is important for the creation of new single-cell organisms and new cells within multicellular organisms. Biologists have come up with the cell theory that states that all cells use cellular reproduction to create new cells and new organisms. These are the two main steps to cellular reproduction. The cell becomes larger, and all of the contents in the cell, including DNA, is replicated. And then cell division, where the parent cell divides into two daughter cells. Because any wrong change in replication could be harmful to the organism, both steps are highly regulated. So we'll start with the most basic thing that our nucleus uses to create us, and also to create the specialized cell. DNA is made of repeating molecules called nucleotides. All nucleotides contain a phosphate, a 5-carbon sugar, and a nitrogen-containing base. The 4 nitrogen bases are adenine, guanine, cytosine, and thymine. Scientists were able to figure out that these always pair up in the same pattern by examining the amount of these 4 bases in many organisms. There was always the same amount of adenine and thymine, and then guanine and cytosine. So these will always pair up together during cellular reproduction, and also when the cell is in a resting state. Human DNA is 31% adenine, 31% thymine, 19% guanine, and then 19% cytosine. From this, scientists discovered that adenine will always pair with thymine, and cytosine will always pair with guanine. So anytime when you see a cellular structure and you see AT and GC, A will stand for adenine, T will stand for thymine, G will stand for guanine, and then C will stand for cytosine. So here is the basic structure of DNA. And you guys can think of DNA as like a ladder. You're going to have your 2 structural frames on the side, and then you're going to have the rungs of the ladder so that you can climb, say, to a roof. So in the main frame of DNA, or the backbone is what is called, you're going to have your sugar and your phosphate backbone. So, and you will have repeating you'll have a repeating sugar that is connected to a phosphate molecule that is then connected to another sugar, connected to another phosphate, so on and so forth. The sugar that DNA uses is called deoxyribose. And then the rungs of the ladder is going to be those nitrogen bases. And again, adenine will always pair with thymine. So here we have one rung, and you'll see AT. So adenine, or the A stands for adenine, the T stands for thymine. Our next rung of the ladder, or two down, is going to be C and G. C standing for cytosine and G standing for guanine. So the nitrogen bases have to be combined together so that the DNA doesn't break and so that there's no damage to the DNA. So the nitrogen bases will be held together by something called hydrogen bonds. Before cellular division, DNA is in the form of something called chromatin. Chromatin is going to contain the DNA and then the proteins that have the appearance of thin threads. I kind of like to think of chromatin as a ball of yarn, and it's going to be in chromatin when the cell is at a resting state. But after, or when the DNA starts replicating, the chromatin is going to start condensing into chromosomes. The condensed chromosomes are going to be a lot easier for the cell to move around than that ball of yarn I like to think of chromatin as. Chromosomes also protect the DNA from any damage during the division process, which is also really important since damaged DNA can create faulty cells, which could lead to cancer. So as chromatin, DNA is going to be wound around proteins called histones. And if you look in the picture here, these little red balls are going to be our histones. The entire DNA thread kind of looks like a string of beads. The beads are going to be called nucleosomes. And so this here is an example of a nucleosome, that DNA wound around the histones. Chromosomes are 10,000 times more compacted than chromatin. Humans have 46 homologous chromosomes, which include 44 autosomes and then 2 sex chromosomes. During DNA replication, the double-stranded DNA is unwound, revealing those nitrogen bases. A new strand of DNA is then created from the parent DNA. These two strands are then connected together, so we end up with two identical DNA strands from our original one. This is called semi-conservative replication. And there are 3 enzymes that are involved in the replication process. You have helicase, DNA ligase, and then DNA polymerase. Unwinding of the DNA is done by the enzyme helicase. Complementary base pairs are joined by the DNA polymerase, and the bases are free floating in the nucleus, and the DNA polymerase grabs the floating bases and pairs them with the bases on the parent strand. The DNA ligase then connects the segment of the sugar phosphate backbone. When the DNA replicates, the entire strand doesn't unwind at one time because the DNA could potentially be damaged. Therefore, the DNA helicase only unwinds section of the DNA one section at a time, and then these are called points of origin of replication. When the helix is separated, it forms a helicase fork. The replication bubble is when the DNA polymerase connects the complementary bases in the replication fork. Replication can only occur in one direction of the DNA strand. It starts with the 5 end and goes until the 3 end. Because the 2 strands are anti-parallel or goes in opposite direction, these replication bubbles move in opposite directions. In this picture, the blue strand is the parent strand, and then the green strand is the daughter strand. The enzyme helicase separates the strands of DNA, creating a fork of sorts. So this will be about right here. With the DNA polymerase connecting the complementary base pairs, you see the replication bubble, and that'll be about right here. You can see that the replication bubble is moving up on the left strand, and then the replication bubble is moving down on the right strand, showing the anti-parallel structure of the DNA strand. And then after the DNA polymerase connects the complementary bases, the DNA ligase connects the sugars and the phosphates together, creating the new strand. And the DNA polymerase is right here, connecting those complementary bases. And then the DNA polymerase is right here, connecting that sugar phosphate backbone. Notice that the green strands are one of the daughter strands of DNA. So both strands of DNA that are created by that parent strand DNA is going to contain one strand of parent DNA and then one strand of the daughter strand. So we're going to have two strands of DNA, and then again each one has one strand of parent DNA and then one strand of the daughter DNA. After DNA is replicated, they are now going to condense into chromosomes. Each daughter strand of DNA are identical to each other and now are called sister chromatids, which this is one sister chromatid, and then this is the other sister chromatid. The sister chromatids are now connected together at a centromere, which is this white dot here. Sister chromatids can easily be confused with homologous chromatids, or homologous chromosomes. Remember that sister chromatids have identical DNA, while homologous chromosomes have the same genes, but different alleles of those genes. About every 7 to 10 years, your body replaces most of its cells. Each of the cells in your body has its own average lifespan. Liver cells last for 10 to 16 months. Red blood cells last for about 120 days. Skin cells are replaced every 39 days. The skeleton is replaced every 10 years. However, some of the cells in your body last your entire life, like your eye lens cells and your brain cells. All these cell cycles have two main steps, interphase and mitosis. About 80% of the cell is going to be in interphase. During interphase, the cell is just doing its thing and will also prepare for division. Interphase, all of the different behavior that the cells are doing in interphase are going to be different for each cell just based on the location of the cell. The last 20% of the cell cycle is the mitotic phase, where the chromosomes and the organelles are separated and then the cell splits. Interphase includes 3 steps: the growth stage 1, also called G1, the synthesis phase, and then the growth phase 2, which is also called G2. Some cells, like muscle and nerve cells, go into a G0 phase, where the organelles and DNA do not divide. They're pretty much just found in a constant state of rest. If a muscle cell is injured, then it will go back into the cell cycle, divide, and repair itself. In the case of nerve cells, they permanently stay in the G0 phase and cannot always repair themselves if damaged. This unfortunately is why brain damage is usually permanent. So during the mitotic phase, also abbreviated to the M phase, a couple of different things occur. First, the cell is going to divide, which is pretty much when all of the organelles inside the cell are going to copy themselves. And then we move on to the division of the nucleus, which is called mitosis. And this is when the parent nuclei are going to copy each other, is going to copy itself. And then afterwards, we're going to have two daughter nuclei. And then after that, we are going to get the division of the actual cell into two daughter cells, which is called cytokinesis. During mitosis, the cell equally distributes the nuclear content of the two daughter nuclei, including the sister chromatids. So here again, we have the original daughter strand of DNA. It is then going to be replicated, and then we have our sister chromatids, which are going to be identical to each other. And the sister chromatids are going to be connected to that centromere. After the nuclei is divided, the sister chromatids will be separated, and one will go into one of the daughter nuclei, and then the second sister chromatid will go into the second daughter cell. When DNA is replicated, there are going to be 92 chromosomes in the nucleus, which is going to be twice the amount of the original DNA that we have in one cell. When the parent nuclei split into two daughter nuclei, each nucleus now contains 46 chromosomes and are identical to each other. Every animal species has an even amount of chromosomes. Another character in the division of the nucleus are going to be spindles. And the spindles are used to split the sister chromatids and were made during the growth phase 2 of interphase. They are made of microtubules, and are created by the centrioles, which are barrel-like structures found in animal cells but not in plant cells. The two centrioles make up the centromeres, which is going to be the primary microtubule organizing center. Centromeres are found in plant cells and still organize the spindle fibers, but you can't see them clearly because they don't have the centrioles. So during mitosis, there are 4 different steps, and a good way to remember them is that they just spell PMAT. So we have prophase, which are this is when the chromosomes are visible under the microscope in condensed pairs. So that's when our chromatin is condensed into chromosomes. We then have metaphase, which is when the chromosomes line up along the equatorial plate, or the middle of the nucleus. And then we have anaphase, which is when the chromosomes are pulled to opposite poles of the cell. Pretty much you can think of when the sister chromatids are separated and then are pulled to the opposite ends of each cell. And then we have telophase and cytokinesis, which are technically 2 different steps, but are more or less blended together since they both happen really quickly. And this is when we have 2 distinct cells visible underneath the microscope. You can actually kind of think of telophase of when those chromosomes arrive to the actual poles of each cell, and cytokinesis is the actual splitting of our original cell, and then this is when the cell membrane are connected on each side of the cell, and then we have 2 fully developed cells. So during prophase, the chromosomes are condensing, each consisting of two sister chromatids held together by a centromere. So you can see here what the DNA would look like uncondensed, called chromatin, and then the DNA then condenses, and notice how thick it is right here. So you can see it a lot better underneath the microscope. And then we have the centrioles here, which will eventually create those spindle fibers about ready to pull the sister chromatids apart. Prophase continues with the disappearance of the nucleolus, which is going to be found inside the nucleus, and then the breakdown of the nuclear envelope, which holds all the contents of the DNA together. The spindle fibers from each pole attach to the chromosomes at the centromere. So during metaphase, the spindle fibers move the chromosomes to the center of the cell, which you can notice here. During anaphase, the sister chromatids are separated and pulled to opposite sides of the cell, which you can already see here that each of our sister chromatids have already started moving to the sides or to the opposite ends of each of the cell. The telophase is when those separated chromosomes reach the edge of the cell, and then cytokinesis happens right after. Cytokinesis is when you can start seeing these dips in the cell, which is when the chromosomes are starting to split. So it's a very distinctive part of mitosis. A very distinctive part of cell division. Although mitosis is divided into different stages, it is a continuous process. Mitosis is usually followed by cytokinesis, again, which is the division of the cytoplasm. During telophase, it begins during telophase and continues after until the daughter nuclei are formed. Plant and animal cells differ. So pretty much during cytokinesis happens mainly in animal cells. Plant cells do not have centrioles, but have similar centromeres. And then animal cells, each centromere has two centrioles. So again, cytokinesis are going to differ in plant and animal cells. Cytokinesis, at least the cytokinesis that we just saw in those pictures, are mainly going to happen within animal cells. And normally cytokinesis is mainly going to be accompanied with mitosis, but not every cell is going to experience cytokinesis. And when they do not experience cytokinesis, but only mitosis, that one cell is going to have two daughter nuclei. This type, certain plants, like fungi, slime molds, and parts of the flowering plants are not going to experience cytokinesis, but instead are going to have two daughter nuclei per cell. As soon as anaphase ends, the cleavage furrow begins to form. A contractile ring is formed from motor proteins called actin filaments, and then pinch the cytoplasm together. The contractile ring will continue to squeeze until the two daughter cells are separated. And you can see this cleavage furrow here, and then another cleavage furrow. And then you can see this line of proteins, which is going to be that contractile ring, until it pinches so far that the two daughter nuclei membranes, until the two cell membranes are fully disconnected. And then we have the cell membrane is then going to be completed on each daughter cell, and so we're going to have 2 fully formed daughter cells. Plant cells have a really strong wall, so cannot form a cleavage furrow. Underneath this electron microscope, as you can see in this picture here, you can see the formation of the cell wall. One of the organelles, called the Golgi apparatus, produces the cell wall components and puts them in vesicles that then travel to the site where the cell is dividing. These then fuse together to make the cell plate, which you can see right here. This new cell plate then releases molecules that will form the new cell wall. This cell plate is later reinforced by stronger filaments called cellulose fibrils and then forms a completed cell wall. So this is mitosis in plant cells. It is going to be the same steps as mitosis in plant cells, but without the centrioles. You can still see the chromosomes condense in prophase, and the lack of the nuclear wall. And then you can still see the spindle fibers and the chromosomes in the center of the cell in mitosis. Here you can still see the chromosomes being pulled apart in anaphase, and then during telophase, the chromosomes have reached the poles of the cell, and then the cell plate is beginning to form. As you can tell from the faint line in the center of the cell, right here, this cell plate is later reinforced with cellulose fibrils and then eventually turns into the cell wall. With animal cells, you can tell it is cytokinesis when you see that cleavage furrow, which is again when that contractile ring is squeezing the plasma membrane together until it separates the 2 daughter cells. And now we'll move on to section 3, which will explain the various steps where the cell kind of make sure that the cell division and nuclear division is happening correctly. This ensures that the stages occur in order and that the cycle continues only when the previous stage is successfully completed. There are going to be several checkpoints. There's going to be a checkpoint in the growth phase 1. There's going to be a checkpoint in growth phase 2, and then there's going to be a checkpoint during the mitosis stage. So notice that the first 2 checkpoints happens during interphase, and then the 3rd checkpoint happens during the mitosis phase. So during the checkpoint, during growth phase 1, the cell is going to make sure that the DNA is healthy before it replicates. If the DNA is healthy, the cell is committed to dividing after this point. If the cell is not healthy, it will then enter into the G0 phase, where the cell tries to repair the damaged DNA. During the growth phase 1 checkpoint, there are proper signals that help the cell get through the growth phase 1 checkpoint. And then if the DNA is healthy, it will then the cell will then be committed to replicating. During the checkpoint in growth phase 2, the cell makes sure that the DNA is replicated successfully. And if the DNA is damaged, it will also try to repair it. So after the first 2 checkpoints, the mitotic phase will then start. The mitotic stage checkpoint is going to be between metaphase and anaphase, just to make sure that those spindle fibers are correctly attached to the centromere so that the chromosomes can successively be separated. So here we have the G1 phase. And again, this just makes sure that the DNA is healthy before replication. If it is damaged, it can then enter into the G0 phase, where the cell will attempt to repair the damaged DNA. If the DNA cannot be successfully repaired, then the cell will undergo apoptosis, which is cell death. So now we have healthy DNA, and then we enter into the synthesis phase, where the DNA is replicated. And then we enter the G2 phase, which is when the cell checks to see if that DNA was successfully replicated. It again just makes sure that it has two daughter DNA and that there are no nitrogen bases missing. And then we enter into the stage of mitosis. And the checkpoint during mitosis is going to be between metaphase and anaphase. Again, just making sure that those spindle fibers are connected to the centromere. And this is to make sure that the chromosomes are successfully split apart and that each daughter nuclei will have the same amount of chromosomes. If you can notice, the center character is DNA. I'm sure, as you have heard, cancer can occur if the DNA is not healthy, and those cancer cells can continue to replicate, eventually causing tumors or causing that cancer to metastasize, which unfortunately can be unhealthy, which can unfortunately cause death. So again, these different checkpoints are just make sure that the cells are healthy. So there are internal and external signals that regulate the cell cycle. Some of the internal signals are protein cyclin and kinases. The levels of cyclin will vary throughout the cell cycle and will drive certain points of the cell cycle. Kinases are responsible for the removal of the nuclear membrane and the condensing of the chromosome using ATP, which is a form of cell energy. So external signals like epidermal growth factor and estrogen stimulate the division of cells. There are also external signals to keep cells from dividing too much, like contact with each other. There is also a limit to the amount of times one cell can divide. This is based on the length of the telomere, which is found at each DNA strand. The shorter the telomere, the less times the cell can divide. This is to ensure the health of the daughter DNA and the cells that are formed. So if the DNA is damaged and does not, and those checkpoint stages are not passed, then the cell can undergo apoptosis, which is programmed cell death and is a necessary part of the health of any organism. Apoptosis is also regulated by internal and external signals. Apoptosis prevents tumors from forming and gets rid of unnecessary parts of an organism, like the webbing between human digits as they grow in the womb, or the tadpole or the tail of the tadpole as it turns into a frog. So during apoptosis, the damaged cell loses structure, rounds up, and the nucleus collapses. Here you can see a healthy cell. The contents of the cell are going to be protected by a healthy cell membrane, and our nucleus is going to be a perfect circle. As apoptosis continues, our cellular membrane is going to lose its structure, and then the nucleus is no longer going to be a perfect circle. As the DNA fragments are going to occur as the chromatin condenses. And now we move on to section 4, where we will talk about the cell cycle in cancer. Again, the cell cycle is regulated by various internal and external signals that will either promote or inhibit the cell cycle. Cancer may result from an imbalance of those internal and external signals. The definition of cancer is that it is a disease of the cell cycle in which cellular reproduction occurs repeatedly without end. So there are two genes that can cause cancer. Proto-oncogenes and tumor suppressor genes code for the signals that regulate the cell cycle. Cancer can form if there is a mutation in these two cells. Proto-oncogenes code for the proteins that promote the cell cycle and inhibit apoptosis. They are often likened to the gas pedal of a car because they accelerate the cell cycle. Tumor suppressor genes, on the other hand, code for proteins that inhibit the cell cycle and promote apoptosis. When proto-oncogenes mutate, they become cancer-causing genes called oncogenes. When tumor suppressor genes mutate, their products no longer inhibit the cell cycle and prevent apoptosis. An external signal, growth factor, will bind to a receptor which activates the proto-oncogenes, causing the cell to divide. This is good when injuries need to heal. The p53 protein is a tumor suppressor protein that checks the DNA for damage during the growth phase 1, so the new cell can have healthy DNA. Any mutations in these two genes would cause cancer. Other things that can cause cancer is the absence of telomere shortening or translocations. And the absence of the telomere shortening will prevent an older cell from dying and will allow that old cell to continue replicating. And then there's chromosomal rearrangements, also called translocations. And translocations are when portions of a chromosome may break off and attach to other chromosomes, causing broken DNA that can lead to cancer cells. Genes that are associated with cancer are the BRCA1 gene and 2, and which are responsible for breast cancer. We have the RB gene, which is responsible for retinoblastoma, which is a tumor in the eye. And then we have the RET gene, which is responsible for thyroid cancer. So now we move on to section 5, where we will learn the different characteristics that a cancer cell will have. Cancer cells lack differentiation, which means that they don't contribute to the body function. Because they can divide uncontrollably, they are going to be immortal. And they also have an abnormal amount of chromosomes in their nucleus. The development of any type of cancer is called carcinogenesis. Notice that this word contains genesis, meaning beginning. Cancer cells also do not undergo apoptosis. As more cancer cells replicate, tumors are formed. Benign tumors are not cancerous, but malignant tumors are. Malignant cancer cells can metastasize, where these enter the bloodstream or the lymph system and can then travel to new areas and form tumors in the new areas. The tumors can form can form new blood cells to nourish themselves, which is called angiogenesis, and this will allow the tumors to continue to grow bigger. Breast cancer begins when a single abnormal cell grows into a benign tumor. Additional changes allow the tumors to become invasive or malignant and invade nearby tissue. Metastasis occurs when the tumor invades the blood vessels or the lymphatic vessels and start new tumors around the body. A mammogram can help find a tumor cell too small to be felt during your annual doctor visits. Thankfully, there are different types of treatment for cancer. This includes either removing the tumor or interfering with the ability of the cancer cells to reproduce. As rapidly dividing cells, they are going to be susceptible to radiation therapy and chemotherapy. And during these two treatments, this damages the DNA or some aspects of mitosis and unfortunately leads to side effects. Homotherapy is designed to prevent cells from receiving signals, which is then going to prevent the growth and the cellular division of the cancer cells. There are certain behaviors which can help prevent cancer. Protective behaviors include avoiding smoking, which accounts for about 30% of all cancer deaths. There is avoiding sun exposure, sun exposure is a major factor in the development of most skin cancers, also called melanomas. Drinking are prone to different cancers, so avoiding drinking too much can help preventing those particular cancers. With a protective diet, weight loss can help reduce cancer risk. Increasing consumption of foods and vitamin A and C can help produce healthy cells. Avoiding salt cured or pickled foods, and then including cabbage family members in the diet.